Screen capture video 7.3. Continuation. I had left off talking about tonicity. Tonicity is the solute concentration of a solution. And these brackets actually mean concentration of. So solute concentration of the solution. And this is usually the solute concentration of the solution relative to a cell. It doesn't have to be. In the above example, we don't have a cell, but it's usually what we'll be talking about in our circumstances. Keep in mind, water moves freely across the membrane, moves both directions, back and forth, back and forth. Some water molecules will be moving in one direction, others will be moving in the opposite direction. That's constantly occurring. If there's a concentration gradient where you have a higher concentration of solute particles, such as sugar, on one side of the membrane than you do on the other, then you're going to have water moving across to try to equalize and bring those concentrations to the same gradient or equilibrium. And so that's something that we look for. When we talk about a solvent is what is dissolving. And in most cases, water is a solvent. However, there can be solids that are solvents, there can be gases that are solvents, there can be other liquids besides water that are solvents. But generally, if something's going to be dissolving in the cells, it's usually in water, because water makes up approximately 3/4 of our body weight. The solute is what is being dissolved. In this case, it's sugar. It could be salt. It could be a gas. Sometimes gases dissolve in water. Usually they do if they're present with each other. We understand carbon dioxide dissolves better in water than oxygen does. And that's one of the reasons why if you have fish, especially those that have higher oxygen need, then you have to move the water to increase the oxygen molecules in it. And we also understand temperature affects the solute of gas in the water, dissolving into the water. Cool water allows more gas to dissolve in it than warm water. In this illustration, you have the link to the image if you want to go and look at it. I have 3 different types of solutions shown with what appears to be a red blood cell placed in it, and also a plant cell placed in the same solution. An isotonic solution is shown here in the middle. That iso means the same, so same tonicity, meaning that when you have an isotonic solution relative to a cell, it has the same solute concentration and therefore the same water concentration across the membrane. If it has the same solute concentration, the water concentration has to be the same also, because for our purposes we're just considering 2 parts to the solution, the solute and the water, and therefore there will be no net movement across the membrane. So over a period of time, the cell does not have an increase or decrease of water, whether it's a red blood cell or a plant cell. In a hypotonic, when you think of hypo is lower or less than, less tonicity, and they're talking about the solution having a lower solute concentration. That's literally what it refers to. So if the solution has a lower concentration of solute, then the cell must have higher concentration of solute. This is the normal for a plant cell. And carbohydrates such as sugars that are being produced in photosynthesis will cause water to be pulled into the cell, and that creates a pressure. It's called turgor pressure. And when the cell has that pressure, it's referred to as turgid, or turgid, usually turgid, but the pressure is called turgor pressure. Animal cells don't have a cell wall to protect them from the increased osmotic pressure. So just a little bit of water moving in will stress the plasma membrane and cause it to burst, and the cell will not necessarily explode like a bomb explodes, but it will rupture and then it will collapse and the cell is destroyed. This is a problem if you're giving a hypotonic solution to a person or an animal. If the solution is too hypotonic, or you give too much of the solution, it can cause the cells to burst and kill those cells and thereby kill the animal or the person. An example of a hypotonic solution would be if you just gave straight water with no minerals or no carbohydrates dissolved in it. The last solution that I have listed here is a hypertonic solution. Hyper means above. So a higher tonicity or higher solute concentration in the solution than in the cell. That is going to cause water to move from inside the cell into the solution. So over a period of time, the cell will shrivel. And if you're talking about an animal cell, such as a blood cell, that's bad for it because these are under pressure, and when they are starting to shrivel and they're being shoved through capillaries, single file, they tend to rupture. So that's not good for them. For a plant cell will lose turgor pressure and the plant will wilt. And that happens when it loses water. Or if you put minerals, salts around it, such as in the summer if you're making homemade ice cream and you dump the salt water out on the grass, it goes into the soil, it actually pulls water across the membrane into the soil, and those cells lose water, your plants wilt, die, and they turn brown. So water tends to move out of the cells into the hypertonic solution. An example of a hypertonic solution, as I already mentioned, would be a salt water solution, or one that's heavily sugared. The salt water solution is a good example. And those can be used to control microbes. High sugar concentrations of candy is one reason why you don't have to refrigerate candy. That tends to pull water from the cells, and bacteria aren't able to grow on it. High salt concentrations will prevent many microbes from growing, not all, but many. So hams are salted, and sometimes you'll see hams hanging in the supermarket, and they're not refrigerated. And they can do that because of the high salt concentration in them. Though, interestingly, there are some bacteria that can handle higher salt concentrations, such as Staphylococcus aureus. And I saw an example of a sample of ham that had Staphylococcus growing on it that had been sent in from somebody that bought it at a store. That has to do with the movement of water across a membrane. Osmosis is the movement of water across a membrane. Anytime something needs help getting across a membrane, but it doesn't require energy, that's called facilitated transport. And facilitated diffusion, facilitated transport are the same thing. Generally, there's a protein that helps. Water can move across the membrane two different ways. Supposedly, it can just move across, and interestingly, we're told that it can make it across this hydrophobic region, which seems strange. I'm not sure if it's attaching to the hydrophobic head and linking across where they're close together. I don't know. But researchers tell us it can do that, because generally that doesn't happen because waters and lipids don't want to mix. But they also know that there's enough movement of water across the membrane. That doesn't explain it all. They claim that there are what are called aquaporins, proteins that allow water to move from one side of the membrane to the other. It does not require ATP, doesn't require energy, in other words. And therefore, since protein's involved, it's a facilitated diffusion or transport. But part of that definition is ATP is not required, no energy is required, and it's facilitated by the presence of a protein. So just simple diffusion, going right across the phospholipid bilayer. Facilitated diffusion, there's a protein that's helping the molecule move across. And again, it's still going from high concentration to low concentration, so it's a downhill process. No energy is involved. Another type of movement across the membrane requires energy, and in general, anytime that happens, it's called active transport. And it requires energy because the particle that's being moved is going from a low concentration to a high concentration, and that's an uphill process. Therefore, energy is required, and that energy is ATP. There are different types of active transports. If you take anatomy physiology, A&P, we'll talk about the sodium-potassium pump. That is a specific active transport, and ATP is required to maintain that gradient across the cells for muscles and nerves to contract properly. Here they're showing an active transport. Molecules at low concentration are being moved across, in a sense pumped across, with the help of ATP being broken down. That allows for energy to be released and for this molecule to change and to move. It's usually a protein molecule, moving these particles across to a higher concentration. There's some specific types of active transport I want to mention. Endocytosis, this is where material is engulfed by the plasma membrane and moved into the cell, hence the endo, which almost sounds like into. If you follow a more Germanic pronunciation of it, into the cell. Cyto has to do with the cell. Here, the endocytosis is illustrated. A lot of times there are receptors on the surface of the cell, and this would be called receptor-mediated endocytosis. And the chemical binds with those receptors that triggers the plasma membrane to what is sometimes referred to as invaginate, or to cause a lobing inside, and then it pinches off and it becomes one, so that you have this vesicle that was once part of the plasma membrane. It has a membrane around it now, and it's enclosing what was on the outside. This would happen if it's difficult to move the material across the membrane any other way. It can be taken in through this process. And an actual electron micrograph shows this part of the membrane that is moving inward, so it can form around the material on the outside and take it in. When the particles are small and hard to see even with a microscope, it's on a molecular or atomic level, it's called pinocytosis, which literally is cell drinking. Phagocytosis, when they're larger particles that you might be able to see with a microscope, such as cell parts or bacteria, it's called phagocytosis, which is literally cell eating. Exocytosis is pretty much the opposite of endocytosis. That's where a vesicle inside the cell will merge with the plasma membrane, and whatever is in the vesicle is released to the outside. It's exiting the cell. It does require ATP, and some vesicle is usually used. So the vesicle contains some kind of a material, some kind of a chemical that may have been produced by the cell. Could be a hormone. Moves to the edge of the cell, and the membrane around the vesicle becomes one with the plasma membrane, and the contents of the vesicle are released to the outside of the cell. They're secreted. Here, supposedly, in this electron micrograph, they're showing insulins being released by pancreatic cells, or the cells of the pancreas during exocytosis. So it's moving to the edge, joins, and then the contents are released. I believe that if we're going to say pancreas cells, the cells plural, so I think that should have apostrophe after the S, but that's not how it came when I copied it. And I think that brings us to an end of the Unit 7 information. I hope you do well on the test.